Hello everyone, am I back? Give me a minute. So can you see? Just a second, why we can't see? Just a second. Can you see me? Can someone write in the chat box? Can you hear my voice? You can't see me now? Even I can't see myself. Just a second. Let me see why the camera is giving the trouble. Hmm. Give me a minute. It's done. I guess it's done now. Everything is good to go now? Can you see me? Can you hear me? Can we start? Can we continue? Write down in the chat box quickly. Now everything is good to go? Okay, let me open this also. Okay. Okay, everything is good now? Okay. So let's continue. So we have started general microbiology in the morning, right? We have started general microbiology in the morning and we have completed first chapter, that is the staining. We have started the second chapter, that is morphology of the bacteria. In the morphology of the bacteria, I taught you that we have to study 5 things, right? Just a second. We have to study 5 things, but as I have completed 4 of them. We have seen the details of the cell wall. We have seen the details of the capsule. We have seen flagella also and fimbriae also. And now I am finishing the last one, that is bacterial spores, and this chapter 2 will be finished, and then we will move on the next chapter. So see, bacterial spores. I'm starting with the topic bacterial spores. Have you heard what are bacterial spores? Does anyone know कि bacterial spore होते क्या हैं? What do you mean by bacterial spore? You know that or you don't know? Listen, have you heard about hibernation? Some animals do hibernation. What is hibernation? मतलब spores are like hibernation only. Whenever the bacteria get unfavorable condition, whenever any bacteria is there and the bacteria is not getting the suitable condition, by unfavorable condition, I mean the bacteria is not getting the food, the bacteria is in starvation, no adequate temperature, no adequate pH. At that time, bacteria forms spores inside them. Spore is non-reproductive stage. Normally, every bacteria divide every 20 minutes. Normally, every bacteria divide every 20 minutes. The binary fission from 1 to 2, 2 to 4, 4 to 6, 4 to 8, 8 to 16. Likewise, it continues. But whenever any bacteria form a spores, it don't divide. It is a living bacteria, but due to the unfavorable condition, it is a non-reproductive stage, right? That is the meaning of the spore. I mean to say spore तभी बनेगा जब unfavorable condition होगी. Learn. Spore is formed only when unfavorable condition is there, right? Now, how many types of spores are there? There are 2 types of spores. Let me explain. एक होते हैं bulging spore, एक होते हैं non-bulging spore. See. Can you see a bacteria here? Can you see a bacteria here? Let me draw the spores inside them. This is a spore. This is a spore. See, here the spore is bulging the bacteria and here the spore is flat. Got it? So this one is known as bulging. Say yes. And this one is known as non-bulging. So basically, whether the spore is making a bulge or it is flat, based on that, it is of 2 types, bulging and non-bulging. First, learn the 2 types of the spores, bulging and non-bulging. You can see this is bulging, this is bulging, this is bulging. This is flat, non-bulging, non-bulging, non-bulging. Right? Now, based on the location. Yes, very correct, Kavita. Now, based on the location. See, this is central. This is central. This is subterminal before the termination. Subterminal. And this is terminal. So, total 6 types of spores are there. I am enumerating. Bulging central, bulging subterminal, bulging terminal. Same with non-bulging. Non-bulging central, non-bulging subterminal, non-bulging terminal. So first you have to see whether the spore is bulging or non-bulging. And then you have to see one of them, whether it is central, subterminal or terminal. Everyone give me a thumbs up. So if any image is provided to you, would you be able to identify the type of the spore? Would you be able to identify? First you have to see bulging hai ki non-bulging hai. And then you have to see the location. Got it? This is how you have to go. See, you can see a spore forming bacteria. You can see spores kaise dikh rahe hain? Here I can see the spores are non-bulging in this particular set. I can see they are non-bulging. Number two, they are central. So you have to comment on two things: bulge, no bulge, and what is the location. So here I will say the final, it is non-bulging and it is central. So it can be of various types, right? Okay, tell me how many bacteria in this world form the spore? There are many bacteria, but only 4 of them form the spore. Only 4. So I'm having again a mnemonic for you. The mnemonic is BSc chemistry. The mnemonic is BSc chemistry. B stands for Bacillus anthrax. Bacillus anthrax and Bacillus subtilis. ठीक है? S stands for Sporosarcina. It's a rare bacteria. I will not teach you this bacteria. देखो नाम में ही है sporo. In the name itself sporo. So spore तो बनाएगा ही. It will form the spore. There are 2 C. One is Clostridia, one is Coxiella. Clostridia and Coxiella burnetii. So there are BSc chemistry 4 bacteria. There are 4 bacteria which form spore. Bacillus anthrax, Sporosarcina, Clostridia, and Coxiella burnetii. Please mark in your notes. Mark the 4 spore forming bacteria. समझ में आया क्या? Let me revise you. From the beginning, this chapter we have completed. We have seen what is cell wall. Okay जी? We have seen. Okay, चलो cell wall तो छोड़ो. You tell me how many bacteria are capsulated. You tell me how many are spore forming, and you tell me how many are motile. I told you the mnemonic of all of them. First, tell me the number. You will say, ma'am, 9 are capsulated. If you include the fungus also, it will be 10. Otherwise, bacteria are 9. Total organisms are 10. 1 is fungus. Spore forming are only 4. Just now I taught you. And motile are 12. Motile are 12. 6 by polar and 6 by peritrichous. So this we have seen. Okay, first learn the number. Now tell me the mnemonic. Capsulated bacteria, how many are there? Yes. Very killer bacteria have pretty nice capsule. Make B double and C double. Count, they are 10 in number. You can see the full form again and again. Right? Spore forming are only 4. The mnemonic is BSc chemistry. This is the mnemonic. Okay, these are the spore forming. Now coming on the motile bacteria, the 6 polar, very polar solution HCl, है ना? And the peritrichous में cute, baby, SLEP, sleep. है ना? This is how you have to learn first the mnemonic. The first step, learn the number. Second step, learn the mnemonic. Third step, say the full form. The most difficult is to learn the full form. But you try. It's not very difficult, believe me. You can do it if you wish. है ना? So please learn. You get many questions out of that. So you will learn. Definitely you will learn the full forms. I will provide you the PDF also separately of the mnemonics. Please learn all the mnemonics. Okay. Now we are done with this chapter. Are you ready to solve the questions? Read the first question. Tell me the answer. Read the first question. Tell me the answer. Which of the following is non-capsulated bacteria? Non-capsulated organism. Tell me the mnemonic first. What is the mnemonic? Mnemonic is yes, very killer bacteria have pretty nice capsule. Make B double, C double. Now have a look on the question. So I guess P for pneumococcus, it is coming in the mnemonic. है ना? I guess meningococcus, it's Neisseria meningitis. Neisseria meningitis ही meningococcus होता है. I guess you know. So it is also coming in the mnemonic. C for cryptococcus. The cryptococcus is fungus. It is also coming in the mnemonic. It is one of the fungus. But Proteus is not coming. So you all are right. But now you imagine if you don't know this mnemonic. Can you solve this question? No way. You cannot solve this question if you don't know the mnemonic. Mnemonics are like your weapons. You are going for a war. In a war without weapons, you cannot fight. If you want to win, you have to be ready with your weapons. You should know how to use it. So you should be able apply your mnemonics. You should be able to do that. Okay. So please learn the mnemonics thoroughly. Okay. So coming on the next question. Peritrichous flagella is seen in. First tell me the mnemonic. Mnemonic क्या होता है? Peritrichous का mnemonic. Say ma'am, cute, baby, sleep. SLEP, sleep. है ना? Now see, they are saying peritrichous flagella is seen in. So which of the following options is coming in this mnemonic? अच्छा, what is other type of polar flagella का mnemonic? This is peritrichous. Polar का क्या होता है? Very polar solution HCl. Now have a look on the option. Vibrio. Vibrio is coming in polar, not peritrichous. है ना? Campylobacter. Campylobacter is also coming there. है ना? And Legionella. Legionella is also coming there. Got it? Only one of them, Proteus, is coming in this mnemonic. है ना? Only Proteus is coming in this mnemonic, so answer is B. है ना? So you have to learn the full form. Full form are confusing. Even knowing the mnemonic, you can be wrong. You can be wrong at many times. So knowing the full form is more important than knowing the mnemonic. Got it? So only one of them. So listen. Let me change this question. If I change, so currently answer is Proteus. Hana, you know the true mnemonic. If I change this question, which of the following is motile? What is your answer? You will say, ma'am, all of them are motile. Answer will be ABCD all. Hana, three of them by peritracheus, one of them by, three of them by polar, one of them is by peritracheus. If I change the question and do it like, which of the following, I mean, all of the following are motile by polar flagella except. So answer again will be Proteus because it is motile by peritracheus flagella. So we can frame the same question in multiple language. The only thing you should know the mnemonics, the full form of the mnemonics. Coming to the next question. Which of the following organelle in the bacteria help in adhesion? What is the organ of adhesion? Is it cytoplasmic membrane? Is it mesosome? Is it fimbriae or lipopolysaccharide? What is the organ of adhesion? Can you tell me what is the organ of adhesion? Yes, organ of adhesion is fimbriae. Flagella is the organ of locomotion and fimbriae is the organ of adhesion. Fimbriae is also known as pili. I told you. Hai. The next question. Can you tell me difference between gram positive and gram negative organisms? Something unique present in gram positive but not in gram negative. Tell me something which is present only in gram positive but not in gram negative. ऐसी एक चीज बताओ जो gram positive में है लेकिन negative में नहीं. I told you the cell wall का structure of both of them. Yes, most of you are right. Please everyone participate. Come on. At max you will be wrong. चलेगा लेकिन participation is important. So yes, teichoic acid. Teichoic acid is present only in gram positive, not in gram negative. So I told you to learn the unique structures. Peptidoglycan present in both of them. And if I ask you, tell me from the same four options, tell me which is common in gram positive and negative. What is your answer in that case? Common क्या है दोनों में? Tell me a thing which is common in both of them. You will say मैम कॉमन तो पेप्टिडोग्लाइकन होता है लेकिन वो तो ऑप्शन में है नहीं. तो पेप्टिडोग्लाइकन इज अ पॉलीमर. उसके दो मोनोमर क्या थे? What were that? Yes, very good. Yeah, very good. So it is N-acetylnuramic acid. है ना? नाम and nag. In that case, answer will become C. ठीक है? The next question. Can you tell me spore forming gram positive anaerobic bacilli? चलो ये question बाद में करेंगे because I have to teach you the anaerobic bacteria. I will come on this question after 5 minutes. ठीक है? Don't worry. I will come on this question. Let me tell you this chapter. After that, जब मैं इसके questions कराऊंगी ना, I will come on this question also. ठीक है? Because it is partial ना, अभी मजा नहीं आएगा. ठीक है? So let me start chapter number 3 for today. Bacterial growth. Can I start? देखो, all the bacteria divide by binary fission. How does the bacteria divide? Bacteria divide by binary fission. One bacteria divide into two daughter cells. You want to see? See. See, everyone see. One bacteria is coming, dividing into two. Yes, see. One bacteria is coming and dividing into two. This is known as binary fission. Normally, one bacteria in 20 minutes, one bacteria divide into two. So this 20 minutes is known as generation time. Normally, there are exceptions, but usually one bacteria divide into two every 20 minutes. Every 20 minutes, one bacteria divide into two. Got it? One bacteria. But there are some exceptions. As high as 14 hours can be there. As high as 14 days can be there. But usually it is 20 minutes. Okay? Usually it is 20 minutes. Okay? So, if I start with one bacteria, for example, so every 20 minutes it divides into two. Every 20 minutes, so from 1 to 2, 2 to 4, 4 to 8, 8 to 16, then 32, 64, likewise it will happen. So in 24 hours, एक bacteria में से कितने बन जाएंगे? One bacteria will lead to 1021 progeny. है ना? So don't learn the number, just I'm telling you. एक bacteria से इतने daughter cells बन जाएंगे. From one bacteria, you can lead to 1021 progeny. Got it? Got it? So you can see this is a bacteria. है ना? It is doing the binary fission. In the binary fission, आपको सिर्फ एक ही question learn करना है. You have to learn only one question. First, nucleic acid divide into two. You can see the nucleic acid is dividing into two. First, the nucleic acid is duplicating, it is dividing into two. First, and after that, the cytoplasm is dividing into two. So you get the question. So first the nucleus duplicate and then cytoplasm duplicate. You have to learn that. It's a PYQ. Okay, so this is the binary fission. What is generation time? The time required by one bacteria to give two daughter cells. So in how much time one bacteria divide into two? That is known as binary fission. Got it? So this that is known as generation time. So ये सब तो ठीक है. Now the most important thing of this chapter. Everyone, please concentrate on your screen. Please, everyone see. See, I'm teaching you bacterial growth curve. Can you see a graph? I will explain this graph to you. Please, everyone, see, you get many questions from this. Make a 5-star over this topic. It's very important. Dekho, I will simplify it. You know, there is culture media, liquid, solid, and liquid culture media. What is culture media? Culture media. In the culture media, we give nutrition to the bacteria. है ना? इसमें मैंने bacteria inoculate किया. तो bacteria get everything. Bacteria is getting protein, carbohydrate, lipid, सब कुछ मिल रहा है. तो बैक्टीरिया डिवाइड, 1 से 2, 2 से 4, 4 से 8, it keep on dividing like that. So we are giving nutrition in the culture media. So see, this is a test tube. Everyone see, this is a test tube in which it is a culture media. I mean, we are providing nutrition to the bacteria. So I have done the inoculation. In the inoculation, I have implanted 3 bacteria. It can be any number. 3 is just a number. It can be any number. Just suppose I have implanted 3 bacteria, inoculated 3 bacteria in that, right? So what I noticed now, after many hours also, they are not dividing. They are still 3. So you may be thinking, ma'am, why they are not dividing? They got everything. They have protein, they have fat, they have carbohydrate, they have mineral, they have vitamin, they have water. Why they are not dividing? They should eat and they should divide. Why? So during the first few hours, when it is not dividing, it is known as lag phase. What it is known as? Lag. You say, no, ma'am, lag aa raha hai. Lag matlab delay. There is a delay in cell division. So during the first few hours, it's always a lag. It is known as lag phase. समझ में आई बात? It is known as lag phase because bacteria is preparing itself for the doing cell division. Inoculation, Elizabeth, inoculation matlab whatever specimen I am having, this is my specimen. My specimen, Elizabeth, it can be blood, it can be urine, it can be stool, it can be saliva, it can be CSF, anything can be my specimen. It contains bacteria. So I take the bacteria from here and implant it into the culture media. This process is known as inoculation. So I'm putting few bacteria in the culture media. That is the meaning of the inoculation. You got it? So inoculation is done. But as soon as I do the inoculation, I'm expecting the bacteria to divide. But bacteria is not dividing. So this is known as lag phase. What is the reason for the lag? Reason क्या है lag phase का? The bacteria is not dividing. You see, ma'am, it's preparatory phase. Bacteria is preparing itself in the new environment. Bacteria is doing the preparation. Now, for cell division, bacteria require many enzyme, many metabolite. Bacteria is gathering all things. So enzyme क्या क्या लगेगा, है ना? Metabolite कौन सा लगेगा? And the bacteria is gathering everything and start cell division after that. So this is lag. So draw on the graph correspondingly. In the lag, you say number do not increase. Number is constant. See in the graph also, it's lag phase. Number is same. But see, the size become maximum. Bacteria is eating and expanding, है ना? It is becoming obese, you can say, है ना? खा पी के मोटा तगड़ा हो गया, हिंदी में कहें तो, है ना? But it is not dividing. Got it? So size is maximum, number is same. Learn 2 properties of lag. Size is maximum out of the 4 phases. Size is maximum, but number is same. That is lag. अब तैयारी हो गई. The preparation is done. Now bacteria will divide anything. It will keep on dividing 1 to 2, 2 to 4, 8, 16, 32, 12, 64 likewise. It will not stop. Anna? So it is known as exponential growth. What it is known as? Exponential growth. Have you read logarithm chapter in mathematics in your childhood? What is logarithm? Logarithm is exponential growth. Exponential growth को कहते हैं logarithm. So this log is logarithm. Log से याद आना चाहिए logarithm. So this is logarithm. Number become maximum. Logarithm. So first lag, then log. Say lag में क्या होता है? Say ma'am, number is same but size maximum. और log में क्या होता है? Reverse. Number is maximum but size again reduce. देखो size again reduce but number become maximum. So please understand lag and log. How many of you got lag and log? What is lag? The first is lag, then log. What is lag? Lag यानी delay. Lag आ रहा है. Lag. So number is same but size maximum. What is log? You see, ma'am, number become maximum but size again reduces. Got it? That is log. After that, now this test tube, this culture media contain a limited nutrition. Some point will come when nutrition start exhausting. It is not unlimited. कभी ना कभी तो खत्म होगा. One point will come. है ना? When nutrition will start exhausting. है ना? So at that time, some bacteria are dividing, they are increasing in number. But some bacteria are not getting nutrition, they are dying. So whatever the number is increasing, the same number is dying. The number which is formed, the number is dying. So they balance each other. Again, the number become constant. In the test tube, it is known as stationary. See, number is constant. Same ka same yahi hai na? It is known as constant. It is known as stationary phase, also known as Plato phase. Stationary or Plato phase. The reason, to constant to yahan pe bhi tha. In the lag also it was constant. Here also it's constant number. But in the lag it was preparing. Here whatever bacteria formed, they are dying. So it is known as number become constant. Say yes. And after that, the complete nutrition get exhausted. Bacteria are only dying. They are not forming. So number become reduced. This is known as decline phase. So third is stationary and last is decline. Say yes. Everyone got it? I'm telling you one unique property of each of them. Lag. है ना? Lag का मतलब होता है delay. Lag. So lag में learn one thing, size is maximum. ठीक है? Log में learn one thing, number is maximum. ठीक है? Stationary में What is stationary? You see, ma'am, nutrition started exhausting. So when the nutrition get exhausted, is it an unfavorable condition? Yes, it is an unfavorable condition for the bacteria. And I told you during unfavorable conditions, spores are formed. So sporulation takes place in stationary phase. Learn S for S. Sporulation takes place in stationary phase because of unfavorable condition. Sporulation hoga and exotoxin also formed. Bacteria will start forming exotoxin. So two things happens. Both of them occur in unfavorable conditions. Sporulation and exotoxin formation. And last, when the bacteria are only dying in the decline phase, involutionary forms are formed. Dead bacteria are known as involutionary forms. So involutionary forms are there. You got my point? So you tell me lag का खास बात क्या है? What is the unique thing in the lag? Say ma'am, maximum size. Log का खास बात क्या है? What is unique in log? Say number become maximum. It's not written. You can write number become maximum. Stationary may learn two unique things: sporulation, exotoxin formation. And decline may its involutional form, मतलब dead bacteria, dead form. Say yes. So you get three type of questions here. Listen, everyone. Number one, you will get a graph. You can get a graph, I mean, and they will make graph like this, and they will ask you to label it, to label the phases. Can you do the labeling for me? Can you do? Let me say this is A, B, C, D. Can you tell me what is A, what is B, what is C, what is D? You can get MCQs on that. Okay. So we all know A is lag phase. है ना? सबसे पहले lag आता है. Number is same. Then B is log. We all can say C is stationary, also known as play 2, also known as play 2. एक ही बात है stationary और play 2. And D is decline. The first question you can get on this. है ना? It's very easy question. I guess everyone can solve it. Everyone can solve it. है ना? Now tell me the reason for all four. Reason for all four. Lag का reason, write down preparation. Preparatory phase. It is known as preparatory phase. Log का reason, write down exponential growth. Exponential growth का reason है ये. Stationary और P2 का the number which is formed, the same number is dying. That is the reason for stationary. And decline ka reason that they are only dying. They are not formed. So in short, write down the reason for all four. The second thing, the second question you get. The third type of question you get here is the unique property of each phase. So in the lag phase, the unique property is that size is maximum. Okay. In the log, the unique property is that number is maximum. ठीक है? In the stationary, two unique properties. Sporulation take place because of unfavorable condition and exotoxin formation takes place. And in the decline, one unique feature, involutionary forms are formed. This is the thing. You get 16 MCQs on this page. How many of you got it? The first is simple labeling. The second is the reason for each phase. The third is the unique properties of each phase. You all are absolutely right and rocking. Got it? Everyone. So you get questions on that. Can I skip? Can I skip this? Can I remove this page? Everyone got it? This is known as bacterial growth curve. Bacteria is growing. Bacterial growth curve. Got it? So this is the thing, right? We are done with this. Now coming on the next topic, you know, bacteria require oxygen for growth. We all require oxygen. Now we are inspiring oxygen and expiring carbon dioxide. So some bacteria also require oxygen. Based on oxygen requirement, bacteria are of 5 types. Based on oxygen requirement, bacteria are of 5 types. Obligate aerobe, obligate anaerobe, facultative aerobe, facultative anaerobe, or microaerophilic. Remove this one. Last one is not important. Just for confusion, don't make confusion. Remove it. So make 5 things. 5. Obligate aerobe, obligate anaerobe, facultative anaerobe, and microaerophilic. These are the 5 type of bacteria based on oxygen. Based on gram staining nahi hai. Based on acid fast staining nahi hai. Based on oxygen requirement. So you will say, ma'am, what is the meaning of all these? What is the difference in all of these? Okay. I guess everyone know what is aerobe and what is anaerobe. शायद सबको पता है ना? You have studied this. Aerobe are the bacteria which grow in presence of oxygen. And anaerobe are the bacteria which grow in absence of oxygen. Does everyone know this? If you don't know, learn that. ठीक है? The first thing. Now you may be thinking, ma'am, okay, we got it. Aerobe, anaerobe, everyone knows. But what do you mean by obligate and facultative? There are 2 type of aerobe, 2 type of anaerobe. Obligate and facultative का क्या meaning है? Obligate का मतलब होता है strict, very strict. कुछ लोग कैसे होते हैं ना? They are very strict in nature. Have you seen some strict people, very disciplined, army people, or they are very disciplined. तस के मस्त नहीं होंगे वो. They are very strict. So obligate aerobe, they always require oxygen. If you don't give them oxygen, they will die. वो बोलेंगे या तो दो या मरेंगे. बीच का रास्ता नहीं, do or die. है ना? It's like that. They are very strict, peculiar. है ना? And obligate anaerobe, they are also peculiar. They grow only in absence of oxygen. If you give them oxygen, they will die. So they are just opposite, है ना? And they are very disciplined. Obligate matlab strict. The word obligate, the dictionary meaning of obligate. Search the dictionary meaning, not science. Dictionary meaning of obligate is strict. है ना? कहते हैं ना, you are very obligate. I mean, you are very strict in nature. है ना? So that is peculiar. That is the meaning of the obligate. Or facultative like हम जैसे लोग, they are adjustive. वो हर environment में they just adjust, है ना? Maximum world is adjustive only. I guess we all are adjustive. So it is not the thing, the people who are adjustive, they don't have their own preference. They also have some preference. But if they don't get that preference, they don't die. They adjust in the reverse environment. You got my point? So these are the meaning of the facultative. Listen, facultative anaerobe, they are actually aerobe. They like to be aerobe. But if you don't give them aerobic environment, they will not die. They will adjust in anaerobic. So facultative anaerobe are preferably aerobe. They are preferably aerobe. And facultative and aerobe are preferably anaerobe. But if you don't give them anaerobic environment, they will adjust in the opposite. So that is the meaning. So maximum world is this only. Maximum bacteria is this only. Got my point? Obligate are few only. I will give the list. Okay? And the last is microaerophilic bacteria. The microaerophilic, they require only 5 to 10% of oxygen, not all. So tell me the 5 categories. Say, ma'am, obligate, facultative, and microaerophilic. Microaerophilic. Obligate aerobe, obligate anaerobe, facultative aerobe, facultative anaerobe. You got the meaning of all of them, I guess, everyone? They grow in presence of oxygen, they grow in absence of oxygen. In reverse condition, they will see they die. Facultative aerobe are actually anaerobe. Facultative anaerobe are actually aerobe. But in reverse condition, they can adjust. They can adjust. Microaerophilic require 5 to 10% of oxygen. Say yes, people, say yes. Got it? Now from this, you have to learn 3 lists. Obligate aerobe, they are 4 in number, learn it. Obligate anaerobe, they are only 3 in number, learn it. Microaerophilic, they are 3 in number, learn it. And remaining all bacteria will fall in this list, don't learn it. Don't learn. Facultative koi nahi puchega exam mein. Nobody is going to ask the facultative. They will be concerned about obligate. Obligate are rare, na? You have to learn that. So learn the obligate, both the list. You can remove this list. I don't know in the notes what I have given. You can do some modification in the notes. So I'm giving you 3 lists now. Listen. The first list is obligate anaerobe. Who are anaerobe? You tell me. Anaerobe kaun hote hain? They grow in absence of oxygen. What if you give them oxygen? They will die. Because they are obligate. So the mnemonic is can't breathe air. Because if you give them air, they will die. The mnemonic is CBA. Or you can learn ABC. CBA. Can't breathe air. Okay? C stands for Clostridia. B stands for bacterioids. And A is actinomycetes. Please learn. Can't breathe air or ABC. Clostridia, say Clostridia, bacterioids or actinomycetes. कल ये निमोनिक में 3 times दिखाऊंगी जब मैं ये तीनों बैक्टीरिया पढ़ाऊंगी. I will teach you all 3 bacteria one by one. So whenever I will teach you that corresponding bacteria, I will show you this mnemonic. ठीक है? Got it? Please learn. So these are obligate anaerobe. Obligate aerobe is not written here. You just make a cross. The list given to you, this is facultative. I do not want you to learn facultative. Nobody is going to ask that. I want you to remove the facultative list and replace it with the obligate one. So obligate aerobe लिख लो. If it is not written in my notes, I'm not sure मैंने notes में क्या दिया है. ठीक है, लॉन्ग बिफोर मैंने नोट्स बनाए थे, तो उसके बाद काफी मॉडिफिकेशन होता रहता है. ठीक है, सो माइनर चेंजेस, आई डोंट थिंक यू माइंड टू डू दैट. ठीक है, ऑब्लिगेट एरोब का लिस्ट लर्न करो. सो द निमोनिक इज, लेट मी गिव द निमोनिक फर्स्ट, उसके बाद मैं फुल फॉर्म बताती हूँ. नेस्टी मतलब नोटोरियस, नेस्टी पेस्ट मस्ट ब्रीथ. द निमोनिक इज नेस्टी पेस्ट मस्ट ब्रीथ, है ना. नेस्टी मतलब नोटोरियस पेस्ट, मस्ट मतलब करना ही पड़ेगा ब्रीथ, है ना. ठीक है, ओके. ओनली 4 आर गिवन इन द नोट्स. वेरी गुड. इफ इट इज गिवन, द सेम मे बी गिवन. जो मैं बता रही हूँ वही दिया होगा. फिर मैंने पीपीटी में नहीं चेंज किया. आई हैव नॉट चेंज इन द पीपीटी. इट्स ओके. लिसन. सो एन फॉर नोकार्डिया, ओके. पी फॉर सूडोमोनास, एम इज माइकोबैक्टीरियम, एंड बी इज बेसिलस एंथ्रैक्स. ठीक है जी. The same is given to you. So please learn. Nesty pest must be. These are obligate aerobe. And finally, learn the last list. Microaerophilic. They require only 5 to 10% of the oxygen. So here also we have a mnemonic. The mnemonic is hell. Hell, the camp is boring. Hell, camp, boring. Camp is boring. You can learn a mnemonic. Hell, camp is boring. So it's Helicobacter, Campylobacter, bhai. I call them twins because they come together. हर जगह साथ में ही आते हैं. So एक और जगह ये साथ में आए थे. Where? पोलर फ्लैजिला की मोटिलिटी, है ना. वेरी पोलर सॉल्यूशन HCl का H और C है. याद आया, वहां पे भी साथ में आए थे. तो हेल कैंप इज बोरिंग, बोर इज बोरेलिया. सो हेलिकोबैक्टर, कैंपाइलोबैक्टर, बोरेलिया. यू गॉट माय पॉइंट. सो आई एम राइटिंग ऑल टुगेदर. सो टेल मी ऑब्लिगेट एनाएरोब, टेल मी ऑब्लिगेट एरोब and tell me microaerophilic. Only the mnemonic. Who will tell me? Obligate anaerobe का mnemonic लिखो. What is the mnemonic? Don't write the full form. Say the full mnemonic. Say the mnemonic is can't breathe air. It's correlating. They can't breathe air. If they breathe air, they will die. What is the mnemonic for aerobe? Nesty pest must breathe. You know what is the meaning of the must? Must मतलब करना ही पड़ेगा. They have to breathe, otherwise they will die. ठीक है? And microaerophilic hell, camp boring. Campus boring, है ना? So try to say the full form. You get many questions. Either you will get a direct question or a big clinical question जिसमें आपको एक clue होगा. There is a obligate aerobe, obligate anaerobe. So these mnemonics will be very useful for you. Believe me, believe me. So can we proceed? है ना? Now this is based on the oxygen we divided the bacteria into these categories. What is the temperature? Based on the temperature, how many types of bacteria are there? Based on the temperature, again we have three categories of the bacteria. Based on temperature requirement. Some bacteria grow below 20 degree. These are known as psychrophile. Some bacteria grow 20 to 40, 25 to 40. What is human body temperature? Human body temperature is 37. We all know. So here, these are known as mesophile. Mesophile come in this category. Mesophile grow in human body. Okay? And the one who grow 55 to 80 degree, high temperature, they are known as thermophile. So psychrophile, mesophile, thermophile, PMT. You can learn the mnemonic PMT. Psychrophile, mesophile, thermophile. Less than 20, 20 to 40, and 50 to 80. You can learn 55 to 80. So psychrophile, mesophile, thermophile. These are the medically important bacteria. No, B, T, you have to say something T. Anyways, PMT, you can learn anything. So yes. ठीक है, pre-medical test, PMT. ठीक है? So can we go ahead? So this is based on the temperature. We are done with this chapter. We will solve some question. Say the answer quickly. Say the answer quickly. Sporulation occurs in which phase? Sporulation occurs in which phase? Is it lag phase, log phase, stationary phase, decline phase? Let me see who is the first. What is the correct answer, people? Yes, I can't see who's the first. There is a row. You all are right. Yes, Shivam, absolutely right. Yes. You all are right. Yes. So correct answer? The correct answer is the stationary phase. If I change the question from sporulation to exotoxin formation, would you like to change your answer or still same? Would you like to change your answer or still would you like to say the same? Exotoxin se replace kar dun to. So yes, it's still same. So sporulation and exotoxin both take place in stationary phase. Stationary phase also known as P2 phase. Please learn that. Okay, yes, it's same. Maximum cell size seen in which phase? Maximum cell size seen in which phase? Yes? So maximum cell size, is it lag, log, end of P2, early stage of decline? What's the correct answer? Maximum cell size seen in, yes, lag phase. You all are right. It will be lag phase. Very good. Thermophile bacteria, PMT का T क्या था? PMT का T. Thermophile bacteria grow at what temperature? Below 20, 20 to 40, 40 to 60, 60 to 80. So actually it was 55 to 80. So 55 to 80 is not there in the option. Go with 60 to 80. It's near. 55 to 80 and 60 to 80 एक ही बात है, right? And if I change it to mesophile, what is your answer now? M कर दूं तो मैं mesophile कर दूं तो आंसर क्या होगा? आंसर हो जाएगा उस केस में 20 to 40. Human body temperature 37. और इसको psychrophile कर दूं अगर तो आंसर हो जाएगा 20. So we can change the question multiple times, right? The next question. Next question in front of you. Please read it. Which of the following is the obligate anaerobe? What is the mnemonic? Obligate anaerobe का mnemonic क्या है? Anaerobe, can't breathe air. Look at the options. Which is coming in this mnemonic? Can't breathe air का C क्या है? Clostridium. So answer is Clostridium. You all are absolutely right. Answer is Clostridium. Okay? Got it ना? Just apply the mnemonic, apply करना सिखा रही हूँ. ठीक है? So correct answer यहाँ तो Clostridium है. I will change one word. तुम समझ गए. Anaerobe को मैं aerobe कर दूं. तो what is the answer now? Obligate aerobe. What is the answer now in that case? What is the mnemonic? Obligate aerobe का nesty pest must breathe. So apply. Options में देखो क्या है? Yes, very good. So you will say pseudomonas. In that case, answer will become pseudomonas. So it is not the thing कि you just learn the options, है ना? तुम question learn कर लोगे. Examiner will change one word और complete answer change हो जाएगा. So don't go for rectification. Never, never. Don't learn it. Understand the concept. One question we have left behind ना, which I told you कि इस chapter के बाद बताती हूँ. I'm going to that question. We have left this question behind. So now we can solve this question also. We have left this question. And the last chapter I told you after this chapter we will solve. Anyways, read it. So now you will tell me a bacteria which forms spore, a bacteria which is obligate anaerobe, and a bacteria which is a gram positive bacilli, which is fitting in all three categories. So write down the three mnemonic first. How many spore forming bacteria you know, write down. How many anaerobic you know, write down. How many gram positive bacilli you know, write down, and search common in that. That will be your answer. So this question is looking very small, but it's very difficult. Anna, frankly speaking. Chalo, we will try to solve that. So how many spore forming bacteria are there? Only 4. The mnemonic is BSC chemistry. This is spore forming bacteria. The 4 spore forming bacteria. How many anaerobes are there? Again 3. Anna, can't breathe air. Okay? And how many gram positive bacilli is there? How many gram positive bacilli is there? Gram staining का classification बताओ. There are 6 gram positive bacilli. The mnemonic is A, B, C, D, L, M. I want you to say the full form of all these and search the common. In सब में common क्या है? Only one thing is common in all of them. What is that? It's Clostridia. It's Clostridia. It's Clostridia. So my answer is Clostridia. How many of you got it? How many of you got it? This is how we have to approach this question. And also, the question is simple, yet the examiner have checked many things. एक छोटे से question से I checked whether you know spore forming, whether you know anaerobic, whether you know gram positive bacilli, and I have checked whether you can make the common or not. ठीक है? So correct answer is Clostridia. Wait. Yes, correct answer is Clostridia. I am changing one word in the question, and entire answer will change. So instead of anaerobic, if I say aerobic, what is your answer now? What is your answer now? So change one of the list. Spore forming are still same. There is no change in that. Gram positive bacilli is still same. There is no change in that. But instead of anaerobic, you have to say aerobic now. What is the mnemonic? Nasty, pest, must, breathe. Now what is common? You have to find out the common thing ना. अब क्या common हो जाएगा तीनों में? Huh? Yes, you people are right, I guess. In that case, you will say, ma'am, B, B, B. So my answer will become Bacillus anthrax in that case. You got my point? So we are changing one word, answer is changing. See the beauty of the question. So this all you can solve only if you know all the mnemonics. If you don't know the mnemonics, forget it. You can't touch this question. हाँ तुक्का मारना हो तो ठीक है, but confidently you cannot do that. You can do that only if you know the mnemonics of all of them. You got the quality of this question. See the level. It's not moderate. It's difficult level question. Looking very small and simple and one-liner question. No, it's not that way. You should know everything to answer that question. You should know 3 lists. At least you should know 3 lists. Okay, Ji? Can we proceed now? Okay. So coming on the next chapter. We are done with 3 chapters. Coming on chapter number 4 now quickly. Okay. Before going to the next chapter, let me tell you something. Now you have only 2 options in your life. Either study now or regret later. The choice is yours. Regret is the worst feeling in this world, let me tell you. Regret is the worst, even worse than failure. If you are failed now, you can forgive yourself. Just suppose you are studying too hard, and you know, the bad of the bad luck, you are getting 149, not 150, or maybe in 40s, or maybe whatever marks you are getting, but you have given your the best shot. More than that, you cannot study this time. But if you are failed now, you will not feel bad. You will feel, I have given my best shot. यार इससे ज़्यादा पढ़ते तो मर जाते. It is in God's hand. It's okay. I have given my best shot. Now I will try again. But if you are wasting your time right now, you are not studying right now, you are not concentrating, you are doing the things you shouldn't do right now, and then you are not getting the marks. At that time, you are failed again, but with regret. You will have regret also. That I wish उस time ऐसा किया होता. वो जो काश word है ना, काश, वो बहुत hurt करता है, pinch करता है कि काश ऐसा किया होता. सबसे बुरा लगता है मन में सोचना कि काश, वो कभी नहीं आना चाहिए. वो काश वर्ड, आई विश, ये वर्ड कभी नहीं आना चाहिए. You should give your best shot. Now rest is in God's hand. You know the passing percentage, 10%, 20%, whatever. It's in God's hand. But you give your best, na? If you give your best, God is always with you. Believe me. ठीक है? चलो. Anyways, going to the next chapter. Can we start the next chapter now? Huh? So coming to the next chapter, culture media, ultra important chapter. We will start culture media now. ठीक है? So culture media. What is culture media? You tell me first what is culture media. Culture media are of 2 types. It can be solid, it can be semi-solid, it can be liquid. Basically 3 types. Solid, semi-solid, liquid. So in all the culture media, whether it is solid, whether it is semi-solid, ठीक है? Or whether it is liquid, they all contain the nutrition. है ना? So basically we are giving carbohydrate, protein, fat, vitamin, mineral, water, everything to the bacteria in this culture media. So I'm having a specimen. This is my specimen. This is my specimen. So from the specimen, I will take a loop. I will take a loop of the bacteria and inoculate either here, or either here. This is inoculation. Inoculation. I'm shifting the bacteria to one of the media. So bacteria will get everything. भाई तुम्हें खाने में क्या क्या चाहिए? What do you eat in your food? You eat everything, na? You eat a little bit of protein, a thali, a little bit of carbs, a little bit of fat. So bacteria also require everything. So bacteria got everything. They keep on dividing every 20 minutes, every 20 minutes, and they form multiple colonies. They form multiple colonies like this. Hai in the here also they form the turbidity, multiple colonies. So you may be thinking, ma'am, why we are wasting our money in giving back nutrition to the bacteria? Bacteria are not good people, na? Why we are giving food to the bacteria? Why? Because ये बकरी को हलाल करने से पहले का खाना है. You got my point? I guess you got it, है ना? So basically, we want to test the bacteria. We want to do the testing of this bacteria. So to test it, we want to increase its quantity. So by giving the food, we are giving the bacteria grow. Once you grow, we will test you. So it's like that. So this is culture media. Got it? This is culture media. Now culture media, all the culture media contain 4 things in common. All culture media. All culture media have water, number 1. That is H2O, HOH. All culture media have NaCl. खाने में नमक सब कोई खाता है ना? Someone जो बिना नमक के खाना खाता है? No. बैक्टीरिया को भी सॉल्ट चाहिए, है ना? उसको भी अच्छा लगता है टेस्ट. So बैक्टीरिया को भी NaCl चाहिए, ठीक है? So we always provide NaCl. We provide पेप्टोन. पेप्टोन में उसको प्रोटीन मिल जाएगा. We provide meat. सारे बैक्टीरिया को हम मीट एक्सट्रैक्ट भी देते हैं. तो मीट में उसको कार्ब्स भी मिल जाएंगे और उसको फैट भी मिल जाएगा, है ना? सो फैट, प्रोटीन, लिपिड, इलेक्ट्रोलाइट, वाटर सब कुछ दे दिया ना. कभी कभी हम उसको ब्लड भी देते हैं, ठीक है? लेकिन अगार से क्या मिलता है? Agar have nothing, zero nutritive value, no protein, no fat, no carbs, no vitamin, no mineral, no electrolyte. So you may be thinking, ma'am, why we add agar then? You are saying it has nothing. So wastage, why we are adding agar to the nutrient medium? Water is providing HOH, electrolyte is providing NaCl, peptone is providing water. So something is providing something. But you are saying agar have zero nutritive value? Yes, agar have zero nutritive value. Still we add it. Actually, agar is a solidifying agent. So please learn, agar has zero nutrition. Agar don't have any nutritive value. है ना? agar में न तो प्रोटीन होता है, न कार्ब्स होता है, न फैट होता है. agar में कुछ नहीं होता है. So why we are adding agar? It is a solidifying agent. है ना? So I told you, na, if there is a liquid media and I want to convert the liquid either to semi-solid or solid. है ना? I want to convert the liquid either into semi-solid or I want to convert it into solid. So I have to add agar. Agar is a solidifying agent. For semi-solid, I have to add 0.5%. And for solid, I have to add 1 to 2%. Let me check these percentage. I think I'm right. But I have to add the agar. So agar is a solidifying agent that solidifies. Say yes. Yes, I'm right. For semi-solid, we have to add 0.5%. And for solid, we have to add 1 to 2%. Say yes. It has zero nutritive value. It is obtained from an algae. The name of the algae is Gelidium species. Okay? Have I told you swarming? What is swarming? I told you some bacteria are PT Usha. They are very fast, I mean to say, very super fast. So once you inoculate them, they start moving and running here and there. And they produce the waves. Have I told you? So just suppose there is a swarming going on agar. I want to inhibit the swarming. So to inhibit the swarming, we have to make it very hard. So bacteria cannot move and produce the waves. Waves नहीं produce होनी चाहिए. Waves नहीं produce होने के लिए we have to increase the percentage of agar. So to inhibit the swarming, you have to do the percentage 6%. So see, I'm telling you 3 things. I'm having a liquid agar. I'm having a liquid culture media. There are 3 questions asked in your exam. If you want to convert into semi-solid, tell me the percentage. If you want to convert into solid, tell me the percentage. And if you want to inhibit the swarming, tell me the percentage. Let me ask the 3 questions. Tell me the percentage in semi-solid. Tell me the percentage for solid, and tell me the percentage for inhibiting swarming. The 3 different percentage. You got my point? So in liquid, if we add 0.5% agar, it will become semi-solid. In liquid, if we add 1 to 2% agar, it will become solid. And in liquid, if we add 6% agar, swarming will get inhibited. It will become stony hard and bacteria cannot move on that. Say people say yes, react, respond. Got it? You have PYQs on these. इस पे PYQ है. इसका meaning ये है. Just learn it. 6% पे भी PYQ है, 1 to 2 पे भी PYQ है, 0.5 पे अभी तक नहीं है, but आ सकता है. So you have PYQs on that. So please learn and highlight the percentage and write down it's a PYQ. You have to learn the percentage. That is regarding the agar. Now coming on the type of the culture media. How many type of culture media in our syllabus? There are 6 type of culture media. How many? There are 6 type of culture media in our syllabus. कितने तरह का होता है? 6. Let me tell you the names first. फिर सबका meaning, definition and examples. You definitely get one question from that. You can say the paper is leaked or whatever you say. हर साल एक question आता है. हर साल, every year you get one question on that. So don't underestimate. है ना? In the next 15 minutes, you are going to secure one question out of 300. Okay? So please mind it and please take it seriously. So there are 6 type of media. ठीक है? What are the 6 type of media? Enriched, enrichment, selective, differential, indicator, and transport. I will explain the meaning and example of each of them. है ना? Most of the students have confusion between the first two कि enriched और enrichment क्या है मैम, है ना? Because the spelling is same, so they have the confusion. But I will remove your confusion. So let's start with the first one, enriched. Let's start with the first one, enriched से शुरू करते हैं. Enriched, riched. देखो नाम में ही है rich. Enrich is rich. Who is rich? Rich क्या होता है? The people who have something additional. उनको कहते हैं rich people. Rich, है ना? So same thing. Achha, how many of you eat the basic food? Basic food means I am telling you the veg thali. I am a vegetarian, by the way, है ना? So veg thali. वो क्या क्या होता है veg thali में? A little bit of rice, a little bit of chapati, little bit of sabji, little bit of dal, little bit of dessert, little bit of salad. So veg thali is basic nutrition which give a little bit of protein, little bit of fat, little bit of carbohydrate, everything, a mixture of all. This is known as basic nutrition. तो बैक्टीरियाज में वेज थाली कौन है? बैक्टीरियाज की वेज थाली है न्यूट्रिएंट अगार. Nutrient agar is the basal nutrition. It provides something, 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 everything. है ना? So most of the bacteria grow on that. Most of us eat veg thali, na? Do you eat veg thali or do not eat? So most of the bacteria eat veg thali. I mean to say most of the bacteria grows on nutrient agar. But तुमने कुछ लोगों को देखा है? They don't eat it. They think we need something special to eat. We don't eat the basal food. We need something extra. In the same way, we have some notorious bacteria. They require something extra to eat. Otherwise, they will not grow. है ना? So they require one of the 3 things. Either they require blood to eat, or they require egg, or they require serum. Blood, egg, serum. Additional, apart from that, ये तो चाहिए ही चाहिए, साथ में ये भी चाहिए. Then they grow. Otherwise, they will not grow. This is known as rich, something additional, something extra. So basically, the meaning of the enriched. Enriched से मुझे याद आता है rich. Rich से मुझे याद आता है something extra. क्या extra? Nutrient agar तो चाहिए ही चाहिए उनको, है ना? They require nutrient agar, but additionally they require one of the 3 things. Not all 3. Mind my words. One of the 3. Either blood, or serum, or egg. One of them. So this additional thing is making them rich. The nutrition is rich. है ना? Blood, serum, or egg. Say the definition. Say this definition. Yes, Kavita. One of the 3 things. Blood, or serum, or egg. Not only this, along with nutrient agar. Nutrient agar to chahiye hi chahiye. They require the veg thali, but they think they are telling they give us something, one thing additional. Then only we will eat. Otherwise, we will not eat. We will not grow. This is enriched. So tell me the examples. You got the definition. Now tell me the examples. Okay. So you tell me, what does blood agar contain? You will say, ma'am, blood agar contain blood. So is it an example of enriched media? Say yes. It is an example of enriched media because it contains blood. So blood agar contain blood. Does chocolate agar contain chocolate? Blood agar contain blood, we know that. Chocolate agar contain chocolate? No. Chocolate agar don't contain chocolate. Chocolate agar contain heated blood. Once you heat the blood, so RBC will dissociate. I mean, the RBC will rupture and become brown color. So the color is chocolatey. The color is chocolatey brown. That's why it is known as chocolate agar. I don't know whether students know or don't know. Blood agar contain blood. Chocolate agar don't contain chocolate. Chocolate agar also contain blood, but it is heated blood. So since it is blood, na heated है तो क्या हुआ है? तो blood. So it is also an example of enriched media. So blood agar, chocolate agar. तीसरा याद करो, Loeffler's slope. Loeffler's slope में serum होता है. So that's why it is also an example. So learn the example. So I taught you 1 media till now out of 6. 1 हो गया. क्या हो गया? Enriched. Enriched. Tell me the definition. Definition is the media which contain additional one of the 3 things. Which 3 things? Say, ma'am, either blood or serum or egg. One of the 3. Such a media is known as enriched agar. And then I will ask you example. So you give me 3 examples. Say blood agar contain blood. Chocolate agar also contain blood, heated blood. And Loeffler's slope, it contains serum. 4th, you can add egg agar. Egg agar contain egg, but this question will not, they will ask. है ना? Question is on blood agar, chocolate agar, Loeffler's slope. तीनों पे PYQ आ चुका है. It is an example of what? All 3 are example of enriched media because they contain something additional. ठीक है? So learn the definition, learn the example. We are done. Say yes. Say yes. We are done with enriched. Out of the 6, enriched is done. है ना? Definition, example. ठीक है? The next 2 I am taking together. Enrichment and selective. ये दोनों एक जैसे हैं. These are diptoed together. Only one difference I will tell you. है ना? Enrichment has nothing to do with enriched. Enrichment is a judwa brother, twin brother of selective. Enrichment and selective are same. Enrich kar nahi hai wo. So what does they mean? Listen, please understand, it's difficult. I'm having a bacterial suspension. This is my specimen. Now the point is that it contains multiple bacteria. Maybe bacteria number 1, 2, 3, 4, multiple bacteria in that. Maybe just suppose I'm drawing the color coding. Maybe it is having some red bacteria, some blue bacteria. It is having some green bacteria. Like that, it is having multiple bacteria. The point is that. Okay? Now I want to grow. This is my solid media. This is my liquid media. I want to grow one of the bacteria, not all. Is it possible? I want to grow one of them. I want to grow only red of them, not blue, not green. I want to grow one of them, not others. Is it possible? You'll say, ma'am, how is it possible? If you inoculate here or here, if you do the inoculation, they all will get the nutrition, no? So they all will grow. Why only one will grow, others will not grow? How is it possible? You are getting my point. How is it possible? So if it is possible, you tell me how it is possible. There are two ways. It is possible, है ना? So what we can do, you can add the stimulatory substance for the red bacteria and inhibitory for the others. Or else, है ना? Again the same. You can add the stimulatory substances for the bacteria of choice and inhibitory substances for the bacteria to be suppressed. ठीक है? So if we are adding stimulatory or inhibitory substances, so if we are doing so in a solid media, you can see this is solid. It is known as selective. Solid is selective. And if we are doing the same in liquid, the liquid is known as enrichment. Enrichment. You got my point? I tried my best to explain you. Hana? So my point is that selective and enrichment both are same. The only point, selective is solid, enrichment is liquid. In both of them, one thing is there. Both of them, one thing is common, that they contain stimulatory and inhibitory substances. Stimulatory for the bacteria of choice and inhibitory for the bacteria to be suppressed. Please everyone respond, not only 2-3 people. Everyone say you got it or you didn't got it. What is selective, what is enrichment? Okay, so here I will give you the examples. Selective and enrichment. The next two we have taken together. Selective and enrichment. ठीक है, इसके बाद 3 और पढ़ाने हैं मुझे अभी. ठीक है? So you see, the next is enrichment. What is enrichment? It is required in the mixed culture. It is required in the mixed culture where multiple bacteria are there. So we are adding the stimulatory substances for the bacteria of choice and inhibitory substances for the bacteria of suppressed, but in a liquid media. Enrichment are always liquid. Please learn the rule, golden rule. Enrichment liquid ही होते हैं. See the 3 diagram. ये तीनों की तीनों diagram देखो, ये तीनों की तीनों liquid हैं. All 3 are liquid. Liquid को कहते हैं broth. So see the suffix broth and see the suffix water. Suffix देखते से समझ में आ जाएगा. Looking at the suffix, you will get it. है ना? Looking at the suffix. So this is broth, this is water. है ना? Selenite F growth में only Shigella will grow, others will not grow because it is having stimulatory substance for Shigella and inhibitory for others. Tetrathionate growth में only Salmonella will grow, others will not grow because it is having stimulatory substance for Salmonella and inhibitory for others. Alkaline peptone water में Vibrio will grow, others will not grow because it is having stimulatory substances for Vibrio and inhibitory for others. You got it? Harsh, you got it? I'm saying take the media, take either solid media or liquid media. In the solid media also, you are adding stimulatory and inhibitory substance. Here also, you are adding stimulatory and inhibitory. So stimulatory for the bacteria of choice and inhibitory for others. So the solid media is known as selective. The liquid is known as enrichment. You got it? Someone was asking, Harsh, others, everyone? So learn the 3 examples. So please, I will give you PYQ on this. Please learn it. There is a PYQ on alkaline peptone water. Alkaline peptone water में कौन grow करता है? Only one bacteria in the world, Vibrio cholerae. Others do not grow. Tetrathionate growth में कौन grow करता है? Only one, Salmonella. Others will not grow. Selenite growth में only Shigella. Learn the combinations, I mean. है ना? I will show you this page again 3 times tomorrow when I will teach you Shigella, Salmonella and Vibrio. कल पूरा revision होगा लेकिन splitted form में. ठीक है? Whenever the corresponding bacteria will come, I will help you in revising again. ठीक है जी? Can we go ahead? So coming on the selective, we are done with enrichment. Coming on the selective media, it is same. Here also we have mixed growth, है ना? And it is a solid media. The only thing, it is not liquid. It is solid. Here also we are adding the stimulatory and inhibitory substance. Got it? So you have 30 bacteria in your syllabus. Yes or no? Tomorrow I am going to teach you all 30, 25, 30 bacteria. You should know the selective media of all of them. Selective media हर bacteria का होता है. I want you to learn the 30 together. So here I have given only 3 examples. But after the culture media and the notes, have a look. I have given you this list. Have I given you this list? This list I'm talking. So this is a list of selective media. Mark the selective media. This list is given to you where the culture media chapter is finished now. जहाँ पे खत्म हो रहा है वहां पे ये लिस्ट दिया है, इसमें 25-30 बैक्टीरिया हैं. सो प्लीज लर्न ऑल ऑफ देम. आई हैव गिवन ओनली 3 एग्जांपल. 3 नहीं लर्न करने हैं, सारे लर्न करने हैं, है ना. हिंदी में कहूँ तो इतना अच्छा याद करो कि नींद में से उठा के भी किसी भी बैक्टीरिया का कल्चर मीडिया पूछूं ना, सिलेक्टिव मीडिया, तुम्हें याद होना चाहिए. If I ask you selective media of only or any bacteria, whenever you are sleeping, still you should be able to say or tell the answer. You have to mug up to that stage, up to that level. You have to. There is no escape. Learn करना ही पड़ेगा. But currently I'm not reading this list because tomorrow I'm going to teach you all 30 bacteria. So whenever I will teach you the bacteria, I'm going to tell you selective media correspondingly there. So after tomorrow's lecture, when bacteriology is completed, I will give you this homework to open this list now, which I have given you in general. Open this list and cross check what something is missing. You will say, no ma'am, 100% we have studied today. So there is no need to study this list right now because you are not aware of all these. Tomorrow I will explain you in detail, na, bacteria by bacteria. Okay, then you will have an understanding, and after tomorrow's session, tomorrow evening, you read this list by your own and you will get everything in that, and definitely you get one question from that. One PYQ, one question pakka isme se. ठीक है. What Kriti is asking, ma'am, the only difference selective media and liquid. Yes, selective is solid, Kriti, and enrichment is liquid. That is the only difference. You are absolutely right. Selective, you have to learn, Kriti, you have to learn selective of all 30 bacteria. But liquid one, enrichment, I have given you only 3 example. You have to learn 3. Liquid सबका नहीं होता. Liquid में we have only 3 example. Learn by heart, only 3. But the selective solid is of all 30 bacteria. You have to learn. Howsoever you learn, I don't know, but you have to learn. ठीक है. So read this list for last time revision, and tomorrow in bacteriology, bacteria by bacteria, I will teach you. Everyone got it? Can we proceed? So we are done with the 3 culture media till now. I taught you what is enriched, I taught you what is enrichment, and I taught you what is selective. Okay, now I'm coming on the 4th one, 4th, 5th, 6th. The next one is the indicator media. What is the next one? The next one is the indicator media. What is indicator? See, this is a culture media. Please understand. It's very easy. This is a culture media. Here is my inoculation. This is my inoculum. I have inoculated a little bit of bacteria here. Okay, a little bit of bacteria here. So what I'm expecting? I'm expecting this bacteria should eat and divide and form multiple colonies. Colonies. What is a colony, by the way? What is a colony? Colony is a collection of multiple millions of bacteria. एक कॉलोनी में millions of bacteria है, so it is visible without microscope. है ना? One bacteria visible to you? Say no. A cluster of bacteria visible to you? Say no. But millions of bacteria, when they are together, they will be visible to you without microscope. For looking a colony, we don't require a microscope. Got it? Now, but what is the problem here? This is my culture media, and this is my inoculum. है ना? I will see the colony when million time it divide. Then the colony will be visible to me. है ना? But I want before that only, I want to know whether the bacteria is dividing or not. I mean to say, as soon as the bacteria divide, I should come to know कि bacteria divide कर रहा है. Colony बनने से पहले ही मुझे पता चल जाए कि हाँ bacteria ने division शुरू कर दिया. Is there a way? कोई तरीका है? Is there a way we come to know that the bacteria is dividing before making a colony? When the colony is visible, everyone come to know yes, bacteria is dividing. But before the colony appears, can we come to know by any method that bacteria is dividing? Yes, there is a way. There is a way. Add some chemical inside this culture media, है ना? That changes color. As soon as the bacteria start dividing, this chemical will change the color and indicate you that the bacteria is dividing. That is known as indicator media. It contains a chemical that changes color. Say yes, people. Say yes. Got it? Say yes. Huh? So yes. So that is the indicator media. Huh? That is indicating you. So learn 2 examples. Examples are important. So first example is Wilson and Blair. Second example is MacLeod. Learn the 2 examples. Wilson and Blair and MacLeod. Wilson and Blair, salmonella will grow. The salmonella grows in Wilson and Blair. So whenever salmonella will grow, here black color will come. Whenever diphtheria will grow, here also black color will come. So दोनों में color तो black ही है. So what is the chemical which is giving this color? See the spelling. See the spelling what I'm saying. I'm rubbing everything. See. Please highlight the spelling. Highlight sulfide changes to sulfide, which is giving black color. As soon as salmonella divide. So this is sulfide to sulfide. And here it is tellurite to tellurium. Tellurite to tellurium. Okay. What lagging? People are writing it's lagging.